Let me start this video with a quick question. Which of the following hormones is actually part of the adrenergic system? Norepinephrine, epinephrine, or acetylcholine? Before you answer, pause for a moment and think about it. And by the end, you'll be confident about the correct answer. In the previous video, we discussed that the autonomic nervous system is a part of the peripheral nervous system. And we also explained that the peripheral nervous system consists of nerves arising from the brain and spinal cord. Since the autonomic nervous system includes both the sympathetic and parasympathetic divisions, it is naturally also composed of nerves that originate from the brain and spinal cord, extend to various organs, and continuously carry information to and from the central nervous system. When we look specifically at the origin of these divisions, the sympathetic nervous system arises from the spinal cord, mainly from the T1 to L2 segments. In contrast, the parasympathetic nervous system originates from two main regions, the brainstem and the sacral part of the spinal cord. From the brainstem, parasympathetic fibers travel through certain cranial nerves, most importantly, the oculomotor, facial, glossopharyngeal, and vagus nerves. And from the sacral spinal cord, parasympathetic fibers arise from segments S2 to S4. Because of these distinct origins, the sympathetic nervous system is described as having a thoracolumbar origin, whereas the parasympathetic nervous system is said to have a craniosacral origin. In the previous lecture, we used an analogy. Think of central nervous system as a main power station or transformer, while the peripheral nervous system as a vast network of wires and cables spreading out from it. As electrical signals travel through wires to reach their destination, similarly, signals from the brain and spinal cord travel through nerves to various organs of the body. Now, if we imagine the nerves of sympathetic and parasympathetic nervous system as these cables, an important point to remember is that there is not a single continuous cable running directly from the brain or spinal cord all the way to the target organ. Instead, the signal travels in steps. For example, suppose a nerve fiber arises from the T1 segment of the spinal cord. As it leaves the spinal cord, it does not directly reach the organ. First, it connects to an intermediate relay station, which you can think of as an extension board. From this extension board, a second nerve fiber then carries the information forward and transmits it to the target organ. Similarly, the nerve fibers of the sympathetic and parasympathetic nervous systems originate from the brain or spinal cord, and on their way to the target organ, they make a synapse at an intermediate point. This synapse occurs in a structure called an autonomic ganglion, which functions much like that extension board in our analogy. The first neuron arising from the CNS is called the preganglionic fiber. It carries the signal from the brain or spinal cord up to the ganglion. At this point, the message is simply passed from the first nerve fiber to another nerve fiber called the postganglionic fiber, which then carries the signal forward toward the target organ. So in both the sympathetic and parasympathetic systems, communication from the CNS to the organs is achieved through this two-neuron chain rather than a single uninterrupted nerve fiber like in somatic nervous system. Now, an important question arises. What is the benefit of having a two-neuron chain in the autonomic nervous system? Why don't we have a single uninterrupted nerve fiber like we see in the somatic nervous system? The two-neuron chain of the autonomic nervous system is not accidental. It provides several key advantages over a single uninterrupted nerve fiber. First, it allows signal distribution. That is, one preganglionic neuron can synapse with many postganglionic neurons in the ganglion. This means a single signal coming from the brain or spinal cord can be spread to multiple target organs or to a large area of an organ, which is especially useful in situations like the sympathetic fight or flight response, where a single signal increases the heart rate, sweating, dilate the pupils, etc. And secondly, in the somatic nervous system, the pathway is simple and direct. For example, if the brain generates a signal to contract the biceps muscle, that signal travels through a single uninterrupted nerve fiber and the muscle contracts. There is very little room for adjustment. In contrast, in the autonomic nervous system, the pathway involves a two-neuron chain. When a signal is generated by the brain or spinal cord, it first travels through a preganglionic fiber and reaches an autonomic ganglion. Because there is a synapse in between, the signal does not act like a simple on-off switch. Instead, it can be adjusted, strengthened, weakened, or modified before it reaches the target organ. For example, when preganglionic sympathetic fibers reach the ganglion supplying the heart, the response depends on the body's state. At rest, fewer postganglionic neurons are activated, resulting in only a mild increase in heart rate. During stress or exercise, the same preganglionic input can activate many more postganglionic neurons, leading to a much stronger response, such as a marked increase in heart rate and force of contraction. This is how the two-neuron chain provides fine control and flexibility in autonomic responses. Now, the next obvious question is, how do these neurons communicate with each other, and how do they finally act on the target organ? To answer this, we must now discuss the neurotransmitters of the sympathetic and parasympathetic nervous system. The neurotransmitter released by all preganglionic autonomic fibers, whether sympathetic or parasympathetic, is acetylcholine. This acetylcholine is released at the ganglion and helps transmit the signal to the next neuron. After the ganglion, the pathway continues through the postganglionic nerve fiber, and this is where the two systems differ. In the parasympathetic nervous system, the postganglionic fibers also release acetylcholine at the target organ. In contrast, in the sympathetic nervous system, most postganglionic fibers release noradrenaline to produce their effects on the organs. So in simple terms, acetylcholine is the common neurotransmitter at the ganglion for both systems. But at the level of the effector organ, the parasympathetic system uses acetylcholine, while the sympathetic system mainly uses noradrenaline. Now that we know which neurotransmitters are released in the autonomic nervous system, the next step is to understand where these neurotransmitters act. They do not act randomly. Instead, they bind to specific receptors present on the target cells. Broadly, autonomic receptors are classified based on the neurotransmitter that acts on them. Accordingly, we have cholinergic receptors, which respond to acetylcholine, and adrenergic receptors, which respond to noradrenaline and adrenaline. Let's start with cholinergic receptors. Cholinergic receptors are further divided into two main types: nicotinic receptors and muscarinic receptors. Nicotinic receptors are present at the autonomic ganglia and help transmit the signal from the preganglionic to the postganglionic neuron. Muscarinic receptors, on the other hand, are present at the effector organs supplied by the parasympathetic nervous system, such as the heart, glands, and smooth muscles, where they produce parasympathetic effects. Next, we come to adrenergic receptors, which are mainly involved in the sympathetic nervous system. These receptors are located on the target organs where postganglionic sympathetic fibers release noradrenaline. Adrenergic receptors are broadly classified into alpha and beta receptors. Most of the organs in our body receive dual innervation, meaning they are supplied by both the sympathetic and the parasympathetic nervous systems. This dual control allows fine modulation of organ functions. The primary role of ANS is to maintain homeostasis, especially for body functions that we do not consciously control, such as heart rate, BP, respiratory rate, pupillary adjustment to light. Imagine if we had to think every time while climbing up the stairs. I better not forget to increase my heart rate. Or consciously remind ourselves at night to dilate our pupils so we can see better and avoid accidents. Clearly, this would be highly impractical. Thankfully, all these functions are autonomic, meaning they operate beyond our conscious control. They are regulated automatically by our sympathetic and parasympathetic nervous systems working together to maintain internal balance. Now let's extend our discussion by bringing drug into the picture. For the sake of understanding, imagine we invent a drug that specifically acts on nicotinic neuronal receptors at the autonomic ganglia. What would happen if such drug stimulated these receptors? Since nicotinic neuronal receptors are present in both sympathetic and parasympathetic ganglia, stimulating them would increase activity in both systems simultaneously. In other words, you'd be pressing the accelerator and the brake at the same time. The net effect on most organs would be essentially nothing. So in organ with dual innervation, stimulating nicotinic neuronal receptors is largely ineffective. Now consider the opposite scenario, blocking these nicotinic neuronal receptors. Since the ganglionic synapse is same in both sympathetic and parasympathetic pathways, a single blocking drug would effectively shut down the entire autonomic nervous system. You would wipe out all autonomic responses in the body at once. We'll discuss ganglionic blockers later, but even now, you can sense that drugs acting at this level are clinically impractical and largely irrelevant. If we truly want to manipulate the ANS, the goal is to selectively enhancing or blocking one branch, either sympathetic or parasympathetic, but rarely both at the same time. If we want parasympathetic effects, we target muscarinic receptors. If we want sympathetic dominance, we use drugs that act on alpha or beta receptors. Now here is an important concept. Because most organs have dual innervation, blocking one branch automatically allows the other branch to act unopposed. In this way, antagonism can effectively mimic the opposite autonomic response. Let's understand this. Consider a patient with an AV block, where conduction through AV node is impaired. To restore conduction, we want a sympathetic effect. Since sympathetic stimulation increases heart rate and AV nodal conduction, one option is to stimulate beta adrenergic receptors, which would certainly improve conduction. There's another, often cleaner strategy. Because AV node also receives parasympathetic input, we can block muscarinic receptors, removing vagal inhibition. This unmasks sympathetic tone, then improve AV nodal conduction, producing a result similar to beta agonist. So clinically, we often have two ways to achieve the same effect. Once you understand this, you have a powerful framework for thinking about ANS pharmacology. In the sympathetic nervous system, there is a special branch in which the postganglionic nerve fibers also secrete acetylcholine instead of norepinephrine. The best and most clinically relevant example of this pathway is the control of sweating. Even though it is the sympathetic stimulation that controls the firing here, at the level of gland, you still see acetylcholine and muscarinic receptor. This is what we call a sympathetic cholinergic system. Keep in mind that the muscarinic receptor are the one in ANS that controls secretion at large. That means it's the muscarinic drugs that can interfere with ANS regulation of secretion. So muscarinic agonists increase the secretion. On other hand, antagonists cause drying of secretion like dry mouth, no sweating, and so forth. Even in this situation, where sympathetic responses control secretions such as in fight-fright situation, a muscarinic blocker would cancel these secretions. So remember, muscarinic drugs always interfere with secretions. So in this case, despite using acetylcholine at the effector organ, the pathway supplying sweat gland is sympathetic in origin, arising from sympathetic ganglia. Let's talk about adrenal medulla. Adrenal medulla is a neuroectoderm derivative. Embryologically, it has the same origin as autonomic ganglion. Just like autonomic ganglion, it receives preganglionic neurons with acetylcholine, and it has nicotinic neuronal receptor. Enterochromaffin cell of adrenal medulla not send out a postganglionic neuron. Rather, they produced hormone epinephrine. Like norepi, it will bind to alpha and beta receptors. Unlike norepi, this is not a neurotransmitter, as it is not released in a synapse. As a hormone, it is produced in the adrenal gland, released in the blood, and work away from its site of production. Imagine a tissue with receptors, and this receptor is innervated. Suppose this is a nerve terminal, and it releases neurotransmitter. As this receptor is innervated, this neurotransmitter go to the receptor and stimulate them. Suppose this is a blood vessel. A hormone also can travel through the blood, and through its distribution, eventually it found its receptors also. Imagine if there is no innervation. Can a hormone still find its receptor? Since it is released in the blood and distributed in the tissues like a drug, the answer is yes. Hormones still be able to find its receptor. How about the neurotransmitter? If there is no nerve terminal, there is no neurotransmitter, because neurotransmitter only work on innervated receptors. Now the question is, epinephrine is work on innervated or non-innervated receptors? The answer is both. On contrary, norepi is work only on innervated receptors only. Beta-2 receptors are generally not innervated. Now tell me, who going to work on beta-2 and who will never reach? So epinephrine work on beta-2 receptor and norepi never reach it. I hope this is clear. Acetylcholine is also a neurotransmitter used at skeletal muscle junction. This is the alpha motor neuron or lower motor neuron. This motor neuron is purely cholinergic. When depolarized, it releases acetylcholine at synapse and act on nicotinic M-type receptor on skeletal muscles. These NM-type receptor are different from NN-type which we found in ganglion.